The last lecture we discussed about modeling process. Modeling process is a process composed by mainly 4 steps, of which the first one is model formulation, the second one is model solution, the third one is model identification, and the fourth is model validation. When we refer to model formulation, we refer to mainly 2 aspects. The first is creating a qualitative model, and the second is converting the qualitative model that has been created in a quantitative model. Qualitative model usually is a conceptual model that can assume any form. The important thing is that the form that the qualitative model assumes is not mathematical one. So usually a qualitative model is a diagram, a conceptual representation. Starting from the qualitative model, we can create the quantitative model, in which the conceptual model, the diagram, is converted into model equation. This equation can be of different types, can be linear, non-linear, can be deterministic or stochastic. The qualitative model that we choose should be good enough for the purpose, and starting from the qualitative model we can we can convert the qualitative description into a quantitative description. After formulating the model, we go to model solution only in the case in which we know all the model parameters. In the case some of the model parameters are unknown, we need to proceed toward model identification. Model identification means that we solve an identification problem in order to get information about the unknown parameter of the model. However, solving an identification problem requires data, and in order to get data we need to perform experiments, and experiments usually need to be specifically designed. After model identification, we can go directly to model solution. Today we are going to introduce a specific kind of modeling procedure that is called compartmental modeling. Compartmental models are a specific class of models that are widely used for studying the kinetics of materials in physiological systems. Compartmental models represent a class of dynamic models derived from mass balance consideration. Dynamic models means that these kind of models are based on differential equation. Thus, first of all, we have to define what we are considering as materials and what we are considering as kinetics. Materials in this case can be exogenous, as for example a drug or a tracer, or can be endogenous, as for example a substrate or an hormone. And the kinetics can include production or distribution, can include the transport from a tissue to another tissue, the utilization of a specific substance, and substrate hormone control interactions. The qualitative model that we consider it is dependent on the purpose. Thus, when we want to represent the load undertaken by the heart during the cardiac cycle, possibly the electrical analog is the best qualitative model that we can choose. In the other case, if we want to represent, for example, glucose kinetics inside the human body, the compartmental model represents the best qualitative model that we can choose. So qualitative model, as we told previously, is a graphical representation, a conceptual representation that is chosen on the basis of the problem that we have to represent. Compartmental models are models in which the main unit is represented by the compartment. A compartment is a quantity of material that acts as though it is well mixed and kinetically homogeneous. So the material inside the compartment should be considered as well mixed and kinetically homogeneous. But what does it mean well mixed and what does it mean kinetically homogeneous? We will see in the next slide. When we refer to a compartmental model, we can associate the compartmental model to several rooms inside a house. If we have a person living in that house that travels from room to room, at any given time this person may be found, for example, in the living room, in the kitchen, in the bedroom. At no time the same person can be found simultaneously in two different rooms. And then entities within a compartment model will exist in well-defined states. So it means that person that travel, person represent a molecule inside the compartment, cannot be found simultaneously in two different compartments, as we cannot find in the same moment the same person in two different rooms inside an house. What can we consider as compartment? For example, a substance such as plasma glucose could be a compartment. And then different substances could be followed in the plasma. For example, plasma glucose, or plasma lactate, or plasma alanine. Thus, in the same experiment, there can be more than one plasma compartment, one for each of the substances being studied. This leads to the consideration that a physical space may actually represent more than one compartment. So it's not mandatory that a physical space has to represent one compartment. So we can have, for the same physical space, different compartments. So compartment is a theoretical construct which may lump material from several different physical spaces in a system. So if we want to equate a compartment with a physical space, this should be dependent upon the system that we are studying and the assumption that we are making about the model. So we cannot always equate a compartment with a physical space. So when we refer to a compartment, we can distinguish between compartments that can be accessible and other compartments that are not accessible for make measurements. So having given the definition of a compartment, we can describe a compartmental model, and a compartmental model consists of a finite number of compartments with specified interconnections among them. The interconnection among compartments represent fluxes of material which physiologically represent transport from one location to another location, or chemical transformation, or may represent both processes. These fluxes of material can be controlled not only by the two interconnected compartments, but can be controlled also by the compartment inside of the compartmental model, thus inside the system. A compartment is a quantity of material that acts as though it is well mixed and kinetically homogeneous. When we refer to well mixed, we mean that any two samples taken from the compartment at the same time would have the same concentration of the substance being studied, and therefore will be equally representative of the substance contained in a single compartment. The other characteristic of the compartment is that the material is kinetically homogeneous, and kinetic homogeneity means that every particle in the compartment has the same probability of taking the path while leaving the compartment. since when we have a particle that leaves the compartment, it leaves the compartment because there are metabolic events related to transport or utilization, and these particles have the same probability of leaving the compartment due to one of these events. So, thus compartmental models allow to reduce a complex physiological system into a finite number of compartments and pathways. Of course, the required number of compartments depend both on the system being studied and on the richness of the experimental configuration. And the compartmental model is unique for each system study, since it incorporates known and hypothesized physiology and biochemistry. As we told when we talked about modeling methodology, we have said that we have always to define assumption on our model. Thus, on the basis of the assumption that we made, the compartmental model will be developed. And a compartmental model may provide the investigator with the insight into the system structure. Thus, a compartmental model is, of course, a kind of system model. The application. What are the applications of compartmental models? Compartmental models are used to describe transport phenomena in the blood. As we can see in this picture, this is an example of compartmental model related to phenylalanine kinetics. Example, we can see in this slide an epidemiological model represents the rate of susceptible individual to the transmissions, infected people and recovered people. And compartmental model may be also used to describe cellular growth. For example, we can have three compartments, one representing the stem cell, one representing the semi-differentiated cell, and one representing the differentiated cell. We can have that quantity inside the compartment of stem cell change in relation to the fact that stem cell can be differentiated into semi-differentiated cell, may die or may be renewed, and the same holds for the other compartments. Compartmental models have a specific representation. The representation is the one that can be seen in the picture. We have, for example, two compartments, and inside the compartment we have a quantity, a mass of a substance compartment. So QI denotes the mass inside the compartment, and the arrows linking compartment I and compartment J represent fluxes into and out of the compartment. All fluxes are necessarily greater than zero, and the compartment has its own volume, distribution volume, may have fluxes coming from the external, may have exogenous input, may have endogenous input, and the concentration of the substance inside the compartment is described as the ratio between the mass of the compartment, QI, and the distribution volume of the compartment, VI. There is also a convention to represent the fluxes. If we have FJI, means that the flux is going from the compartment I to the compartment J. Vice versa, if we have Fij, means that the flux is going from the compartment J to the compartment I. It means that the first subscript represent the compartment that receive the flux, whereas the second subscript represents the flux from which the flux derives. But Pi represents an endogenous input. It means a flux related to de novo synthesis of material. Instead, Ui represents an exogenous input. As I previously described, fluxes are represented with a specific convention in the notation. If we have F0I, it means that the flux comes from the compartment I and is going to the environment, thus out of the system. At the same time, Fji means a flux from compartment I to compartment J. Fij means a flux from compartment J to compartment I. Starting from this model, that is a conceptual model, we can write model equation. Thus, we can go from a qualitative description to a quantitative description. The general equation for the compartmental model are obtained by writing the mass balance equation for each compartment. It means that the law that holds for this kind of model is a mass balance equation. For what concerns the mass balance equation, we have that the variation of the quantity inside the compartment I is given by the sum of the fluxes entering the compartment I minus the sum of the fluxes coming out from the compartment I. In this description are included the fluxes between compartments, but are included also endogenous and exogenous input. Each compartment is also described by its initial condition, and initial condition for the compartment I is represented as Qi at time 0 equal to Qi0, where Qi0 denotes the mass in the compartment at time 0. In this equation, that is a mass balance equation, we have that fluxes coming into the compartment are highlighted with the orange circle. We have all the fluxes coming into the compartment. Sum of the fluxes Fij, thus the fluxes between the compartment I and the other compartments. Then we have the endogenous input Pi and the exogenous input Ui, all the fluxes coming into the compartment. And now we can see all the fluxes going out from the compartment. Thus we have all the fluxes coming out from compartment I and going to other compartment, as for example Fji, and then we have all the other output, thus for example F0I, the flux that is going out from compartment I to the external. Usually the fluxes are function of time and all compartmental masses. Thus we have that each flux can change in relation to time and in relation to the quantity inside the compartment of the model, as can be seen from these equations. If we consider an accessible compartment, we can measure concentration of the material inside the compartment. Thus, concentration of the material inside the compartment is represented by Ci as a function of time equal to the ratio between Qi as a function of time and Vi. This is called the measurement equation, and this holds only for compartments that are accessible